Hi guys, good evening.
Good evening, sir.
How are we?
I'm great. How are you, sir?
I'm okay as well. Okay, so welcome each and every one of us to our session for today. And we are looking at, we're continuing looking at cell division. And today we are looking at meiosis to be specific. We are continuing looking at cell division. And basically we are looking at meiosis. So meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. So last time we did look at the cell cycle. The cell cycle, those that remember, what did we say the cell cycle is? Guys, what's a cell cycle? We've forgotten. We said the cell cycle is actually a series of events. It's a series of events that takes place in a cell, which leads to the cell's replication or duplication. So a cell cycle is a series of events which takes place in a cell, leading to its division and replication or duplication. That's what we talked about. And the picture on your right summarizes what the cell cycle is, summarizes what the cell cycle is. We did mention that in eukaryotic cells, the cell cycle is divided mainly into two periods. Two periods. So we have the interphase and the mitotic phase, or the M phase. So you have the interphase here, the interphase, and the M phase, which comprises the cell cycle. So the M phase, the interphase, and the M phase. And we said that the interphase is actually the preparatory phase which takes place before the M phase. And it's important during this phase that the cell synthesizes the different molecules, the different proteins, the different enzymes, which will be shared with the new daughter cells. And this stage of the interphase stage, you say that it's divided into the gap phase, which is G1, and then in the synthesis stage and in the G2 or gap 2 phase. So we talked about this, which is the cell cycle. And mitosis and meiosis, we did mention that actually the major types of cell division that occur in eukaryotic cells. And then at the end of the lesson last time, we did mention that meiosis is a type of division or cell division which results in the production of gametes. It results in the formation of gametes, and these are either sperms or ova, so known as eggs, and these occur in sexual reproduction. They occur in sexual reproduction. So let's look at meiosis. Let's look at meiosis. So we are saying that meiosis is involved in the production of the sperm and eggs or ova in individuals or organisms that can sexually reproduce. So after this process has taken place, we see that the chromosomes will be reduced by half, to be reduced by half. So in the case where a sperm fertilizes an egg, that zygote that result from the process of fertilization, we actually have a full set of chromosomes. So a cell that undergoes meiosis, therefore, is going to divide twice. That is meiosis 1 and meiosis 2. So it means that the diploid, which is a 2N parental cell, will result into 4 haploid gametes. So haploid, which is designated as an N. So diploid from the parent will result in a 4 haploid gametes. So we said that meiosis is divided into meiosis 1 and meiosis 2. So meiosis 1 is known as it's known as the reduction phase. Reduction phase because meiosis 1 reduces the number of chromosomes from 46 chromosomes or 2N to 23 chromosomes or N, just as we have mentioned there. So it's going to reduce the number of chromosomes from the diploid, that is for 2N or 46 chromosomes, to the haploid or 23 chromosomes. So now that's called the reduction phase. While meiosis 2 is the division phase. So in meiosis 2, it's the equatorial analog of mitosis. Meiosis 2 is the equatorial analog of mitosis. So we are saying in meiosis, unlike what happens in mitosis, 2 chromosomes in a homologous pair are going to line up next to each other, and that's what is known as, leading to a formation of what is known as a synapsis. So the resulting homologous pair is referred to as a bivalent. The resulting homologous pair will be referred as a bivalent. So we're going to meet this word as we go along. So why is meiosis important? Why is meiosis important? So meiosis is very important in that it leads to the maintenance of a constant number of chromosomes in a given species. It leads to a constant number of chromosomes in a given species. So that's to ensure that the number of the chromosomes is reduced to half during that gametogenesis or the gamete formation, so that the total number of chromosomes for that species will actually be maintained, so it will remain constant. The other thing is that through reduction or through meiosis, there is production of genetic variation. So recombination and random orientation takes place. So recombination and random orientation of the chromosomes takes place during the process of meiosis. So this process further enhances the process of fertilization. Therefore, this process of meiosis or reduction division leads to genetic variation. And this is very important in the long-term survival of the species. It's very important, the long-term survival of the species. So interphase, the process. So we start looking at the process of meiosis. So we start with the interphase. So like we said, the interphase is actually similar to that of mitosis, except for the fact that in meiosis, interphase is followed by 2 cell divisions. So the same way interphase takes place, or similarly as in mitosis, also in meiosis, it takes place in the similar manner, except that in meiosis, we have meiosis 1 and meiosis 2. So now let's look at meiosis 1. Meiosis 1. So we said when we look at meiosis, It is going to thus involve a single chromosomal replication and two divisions of the cytoplasm. So we are going to have cytokinesis 1 and cytokinesis 2. And chromosomal replication in meiosis actually takes place only during the interphase phase, just before meiosis 1 starts to take place. So meiosis 1 actually consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. So it consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. And of course, we have cytokinesis 1 as well. So the first stage is the stage of prophase 1. So what happens during the during prophase 1? What happens during prophase 1? So what will happen during the process of prophase is that the double-stranded chromosomes are going to condense. In prophase, they condense. So generally, when we look at the stage of prophase, we're going to see that the homologous chromosomes are going to condense. And then these homologous chromosomes are actually paired. The homologous chromosomes are paired. And then there's recombination of genetic material which occurs between the maternal and paternal chromosome pairs. There is a recombination of genetic material between the maternal and paternal chromosomes. So when the homologous pairs lie side by side, they lie in a pairing that leads to a formation of what is known as a synapsis. So they become a pair known as the synapsis, and each of those homologous pairs will actually be known as bivalent. And as we mentioned already in the preamble, we talked about bivalent, which means simply two chromosomes. So when you look at meiosis, the meiotic prophase 1 is subdivided into five stages. Subdivided into 5 stages. We have the first stage, which is known as leptotin. We have the first stage, which is known as leptotin. So during leptotin, what happens is that there's condensation of chromatin, and also there's the appearance of chromosomes. So condensation of chromatin happens during the stage of leptotin. So this is the first stage of prophase 1, leptotin. And then after leptotin, we have what is known as zygotin. We have zygotin, in which synapsis takes place through the close association of the homologous chromosomes. So synapsis begins at this stage, and it continues through to the next stage of throughout the next stage of patching. So synapsis starts to take place through close association of the homologous chromosomes during the zygotin stage of mitotic prophase 1. Mitotic prophase 1. So during the synapsis, we have non-sister chromatid of the tetrad, which can cross over and also exchange portions. And this is what takes place during the process of, or during the stage of patching. During the stage of patching, there's crossing over which occurs, and this involves transposition of DNA strands between the two homologous chromosomes, the two homologous chromosomes are going to, there will be exchange of genetic material through a point known as the chiasma to the process or point known as the chiasma. So this is a chiasma or chiasmata when it's plural. So remember we mentioned genetic variation as being one of the important functions of meiosis. So crossing over that happens during the process of prophase 1 and patched in there, as we have seen during this process. It's important, or rather crossing over that takes place during this process of prophase 1 is actually the first major source of genetic variation. And during diplotting, we see that the homologous chromosomes begin to separate from each other and they appear to be connected by newly formed junctions between the chromosomes. And as I mentioned, this is what is known as the chiasmata. So chiasmata is plural. Chiasmata is plural for chiasma. And then we have the dikinesis stage. So during this stage, we see that the homologous chromosomes are going to condense and shorten. And so during this stage, they reach their maximum thickness. And also the nucleolus is going to disappear and the nuclear envelope is going to disintegrate during the process of the dikinesis, in the process of dikinesis. So this is what happens in prophase 1. This is what happens in prophase 1. And these diagrams are summarizing what happens during this prophase 1. And then after prophase 1, we have metaphase 1. We have metaphase 1. So during the metaphase 1, what will happen is that the tetrads are going to line up along the equator. So they're going to line up along the equator. So they have lined up along the equator. So those divalent, the bivalents are going to align themselves in actually an orderly, though random manner at the equatorial plate. So at the equatorial plate, they are going to line up. And then we see, that during the metaphase 1, the spindle fibers are going to attach to the centromere region of each of these homologous pair. And then we have other metaphase events occur as the same that happens in mitosis. So in the late metaphase, the chiasmata are going to be cleaved and the chromosomes are going to separate. Are going to be cleaved and the chromosomes are going to separate. So this is what happens in metaphase 1. Metaphase 1. The bivalents are going to align themselves in an orderly, though random manner on the equatorial plate. They're going to line up on the equatorial plate. And it's very important that there is this random orientation of the chromosomes, that there's this random orientation of the bivalents, as this promotes independent assortments of the chromosomes and their genes as well. And this leads further to addition of genetic variation as well. So that's metaphase 1. And then we have anaphase 1. We have anaphase 1. So during anaphase 1, what is going to happen is that the chromosomes are going to start moving to the opposite sides or opposite poles of the cell. They're going to move to the opposite cell or opposite pores of the cell. So anaphase 1, the tetrads, in short, are going to separate the tetrads or bivalent. Remember that each chromosome has 2 chromatids. So hence a bivalent has actually 4 chromatids and is therefore also known as a tetrad. So in anaphase 1, we see that the tetrads are going to separate. And when they separate, they are drawn to the opposite pores by the spindle fibers. They're going to be put to the opposite pores by the spindle fibers during this process of anaphase 1. So the centromeres are going to remain intact during this event of anaphase 1. So the microtubules and the kinetocoh fibers also interact and there is movement. So a maternal or paternal member of each homologous pair now containing exchange segments are going to move to each pore. Remember, there was a genetic recombination through the crossing over that has happened. The members were able to exchange genetic information. So no wonder there's this statement of containing exchange segments. They are able to move to each pore. So unlike in mitosis, we see that the sister chromatids are during the process of anaphase 1 are going to remain together, So we see that each pore is going to receive a haploid number of chromosomes. And these chromatids, remember these 2 chromatids are actually recombinant. They are actually recombinant chromatids because of the exchangement of the segment that happened during crossing over. So this is what happens in anaphase 1. So after anaphase 1, we have telophase 1. We have telophase 1. So during telophase 1, the spindle fibers are going to continue moving homologous chromosomes to the pores, after which either pore has a haploid number of chromosomes. So each pore will have a haploid number of chromosomes. There'll be a haploid number of chromosomes. So in telophase 1, we see that the separated chromosomes are going to arrive at the opposite pores. And then this movement that happens leads to the reduction of the chromosomal number from 2n, which is diploid, to n, or which is known as haploid. And then we see that the nuclear membrane is also going to reappear and also to surround each set of membrane. It's going to, or rather, surround each set of the chromosome. So we see the nuclear membrane reappearing. And then the chromosomes are also going to uncoil back into chromatin, chromatin during the process of telophase 1. And so cytokinesis 1 is going to take place. Cytokinesis 1 is going to take place. So the diploid cells are going to divide into 2 haploid cells through a cleavage furrow. And remember, a cleavage furrow is in animals or a cell plate in plants. So the 2 daughter cells that are formed, each contains half number of chromosomes as the original parent. Remember I said that this is the reduction phase. So after that, the cell now is going to undergo meiosis 2. So the cell will quickly enter into meiosis 2 without passing through an S phase. And like I mentioned, that meiosis 2 is an equatorial division and resembles mitosis. It's an equatorial analog of mitosis. So we have during interphase 2, what will happen is that the cell is going to rest between meiosis 1 and meiosis 2. And no DNA or chromosomal replication takes place during the interphase 2. So the cell now goes to prophase 2, which is the first stage of prophase 2. So during prophase 2, the nuclear membrane as well as the nuclei are going to break and the spindle fibers are going to reappear. So the nuclear membrane disappear, the spindle fibers appear, chromatin is also going to thicken and also shorten. So the chromosomes then start migrating to the equator of the cell. So the chromosomes, remember I said that the chromatin is going to thicken and shorten. So the chromosomes will condense again, following a brief interphase in which DNA does not replicate, as I mentioned earlier. So this is prophase 2. So during prophase 2, there is no synapsis, there is no crossing over that takes place. Remember in prophase 1, there was synapsis and crossing over that took place. In prophase 2, there is no synapsis and there's no crossing over that happens. And then we have metaphase 2. Metaphase 2. So during metaphase 2, we see that the chromosomes, as we can see, the chromosomes are going to line up. The chromosomes are going to line up at the second metaphase plate at the center of the cell. And then the kinetocores are then going to point towards the opposite poles of the cell. So chromosomes line up in an orderly manner along the metaphase plate. And then also what will happen, as we can see, we see that the new metaphase plate is actually rotated by 90 degrees perpendicular to the metaphase plate. So during metaphase, we see that the chromosomes line up at the second metaphase plate at the center of the cell. And then after metaphase, we are going to anaphase 2. So remember in metaphase, we said the spindles are going to move the chromosomes into an equatorial area and also attaching to the opposite sides of the centromeres in the kinetochore region. And so when anaphase 2 comes, the centromeres are going to split and the former chromatids, or otherwise known as the chromosomes, now are going to segregate into opposite sides of the cell. So the sister chromatids are going to divide and move to the opposite poles of the cells. The sister chromatids are going to divide and move to the opposite poles of the cells. So they are going to separate and become chromosomes in their own right and are put to the opposite poles during the event of anaphase 2. And then after anaphase 2, rather, we have telophase 2. Telophase 2 taking place. So in telophase 2, the chromosomes are going to uncoil. They are going to uncoil during the process of telophase 2. They are going to uncoil and they are going to lengthen into chromatin. They're going to uncoil and lengthen into chromatin. And then the spindle fibers are going to disappear and the membrane is going to reform. So in this phase, we see that distinct nuclei start forming at the opposite pole, and cytokine starts forming. So here we have telophase 2 and cytokinesis 2. So the cytokinesis is going to separate the cells. Cytokinesis is going to separate the cells. So the overall result of meiosis is the production of 4 daughter cells. So 4 daughter cells are going to be produced. And each of these daughter cells will contain a haploid number of the chromosomes. It's going to contain the haploid number of chromosomes. So it's going to contain N instead of the original 2N of the parental cell. So at the end of the process of meiosis, the product is 4 daughter cells having haploid number of chromosomes. So this table is simply summarizing what we've talked about. We've talked about prophase 1, where the chromosomes are going to condense, the nuclear envelope is going to fragment. We see the homologous chromosomes binding firmly together along their length, and forming a tetrad or a bivariant. And we see formation of the chiasmata between the non-sister chromatids, and also crossing over happening during the stage of prophase 1. And we see the spindle fibers emerging from the central subs. And then we also have a process known as a pro-metaphase 1. A pro-metaphase 1 in which the homologous chromosomes are attached to the spindle microtubules at the kinetochore or the fused kinetochore shared by the sister chromatids. And so we see that during the process of pro-metaphase 1, the chromosomes are going to continue to condense, and we see the nuclear envelope completely disappearing during pro-metaphase 1. And then we talked about metaphase 1, in which the homologous chromosomes are randomly assembled at the metaphase plate, where they have been maneuvered into place by the microtubules. So the microtubules are going to help move or maneuver into this place, into the metaphase plate. We also talked about anaphase 1, in which we saw the spindle microtubules pulling the homologous chromosomes apart, and we see that actually the sister chromatid will still remain attached to the central. So I'm doing the anaphase 1, doing the anaphase 1. And then we saw during telophase 1 that the sister chromatids are going to be, the separated chromosomes or sister chromatids are going to arrive at the opposite pose. They arrive at the opposite pose and they begin to decondense. And we see a nuclear envelope starting forming around each nucleus, and the cytoplasm being divided by a cleavage furrow or a plate in plants and in animals and plants respectively. And we see what results are 2 haploid cells. So each cell contains 1 duplicated copy of each homologous chromosome pair. We did look at prophase 2. We saw that during prophase 2, there's no crossing over that happens. There's no synapses that happens. We see that during prophase 2, the sister chromatids are going to condense. So chromatin, in short, thickens and shortens, and we see a new spindle fiber starting to form. And then also the nuclear envelope disappears, or it begins to disintegrate or fragment. So during pro-metaphase 2, the nuclear envelope is going to disappear, and the spindle fibers are going to engage the individual kinetochores on the sister chromatid, such that when the cell reaches metaphase 2, the sister chromatids are going to line up at the metaphase plate, are going to line up at the metaphase plate. And then during the process of anaphase 2, we see the sister chromatids being pulled apart by the shortening of the kinetochore. By the shortening of the kinetochore, they are going to be pulled apart. And then the non-kinetochore microtubules are going to lengthen the cell. They're going to lengthen the cell. And then we have the process of telophase 2 and cytokinesis, in which we see that the chromosomes having arrived at the pores and having have to decondense, and we see the nuclear envelope developing and surrounding the 4 nuclei, and we see the formation of the cleavage furrow. See the formation of the cleavage furrow, which divides the 2 cells into the 4 haploid cells, which are the results or products of meiosis. So this is the process of meiosis. This is how meiosis takes place. So having reached at this stage, okay, just log out and log in back so that we can summarize and we end the session for today.